Approximately 600 million years ago, Earth bore little resemblance to the world that exists today. The continents were arranged in configurations that modern geography cannot easily map. Landmasses clustered near equatorial and southern latitudes, separated by shallow epicontinental seas that extended across vast stretches of the planet's surface. The atmosphere held oxygen concentrations significantly lower than present levels, estimated at roughly 10 to 15% by volume, and the oceans, though chemically stratified, were beginning a slow but measurable shift toward greater oxygenation in their upper layers. This was the Ediacaran period, a span of approximately 96 million years, running from around 635 to 539 million years ago, and it represents the earliest chapter in the history of complex macroscopic life on Earth. The planet had only recently emerged from one of the most severe glaciation events in its history. Evidence preserved in sedimentary sequences across multiple continents indicates that, prior to the Ediacaran, ice sheets had extended to equatorial latitudes on at least 2 separate occasions, a condition referred to in current geological models as snowball Earth. The termination of this glaciation, driven by the gradual accumulation of volcanic carbon dioxide in the absence of ice-free silicate weathering, produced a rapid greenhouse transition. Global temperatures rose, ice retreated, and in the oceans that followed, conditions began, slowly and unevenly, to support life of a kind the planet had never produced before. For the first several billion years of Earth's history, life had existed exclusively at the microbial scale. Prokaryotic organisms, bacteria and archaea, had dominated the oceans, since at least 3.5 billion years ago, forming layered microbial mats, processing the chemistry of the early atmosphere, and slowly, over immense spans of time, altering the conditions of the planet itself. The accumulation of free oxygen in the atmosphere, beginning in earnest around 2.4 billion years ago during the Great Oxidation Event, was a direct product of this microbial activity. By the time the Ediacaran began, this process had continued long enough that oxygen was present in sufficient quantities to support a new tier of biological complexity, organisms composed of many cells, capable of growth at scales visible to the unaided eye. The earliest evidence for these organisms appears in the fossil record at approximately 580 million years ago in a geological assemblage known as the Doshantuo Formation in what is now southern China, and in the Ediacaran assemblages of the Flinders Ranges in South Australia. What these fossils reveal is a biosphere unlike anything that followed it, a world populated by soft-bodied organisms of uncertain classification, many of which bear no clear relationship to any animal phylum currently recognized. These organisms are grouped collectively under the term Ediacaran biota. They were, as a rule, entirely soft-bodied, possessing no mineralized shells, no hardened skeletons, no calcified structures of any kind. Their preservation in the fossil record is itself remarkable, a consequence of rapid burial in fine-grained sediments and, in many cases, the presence of microbial mats on the seafloor that facilitated the formation of impressions before decomposition could erase them. Without these conditions, the record of Ediacaran life would be largely invisible. The forms themselves were extraordinary in their variety and their strangeness. Dickinsonia, one of the most studied Ediacaran organisms, was a bilaterally symmetrical, oval-shaped body, divided into a series of parallel segments, ranging in size from a few millimeters to approximately 1.4 meters in length. It lay flat against the seafloor or microbial mat surface, and chemical analysis of exceptionally preserved specimens has detected traces of cholesterol, a molecule associated with animal cell membranes, suggesting it was in some form an early animal. Yet its precise phylogenetic position remains a subject of scientific debate. It had no apparent mouth, no digestive tract, no appendages. Current hypotheses suggest it may have absorbed nutrients directly through its lower surface in a manner consistent with osmotrophic or diffusion-based feeding. Charnia was another characteristic form, a frond-like organism resembling, in superficial appearance, a sea pen, though the resemblance may be entirely convergent. It projected upward from the seafloor, its body organized into a series of branching units arranged along a central axis. Specimens have been recovered from deep-water sedimentary deposits, suggesting that Charnia and similar frondose organisms were capable of surviving in low-light, low-energy environments, possibly filter-feeding from organic particles suspended in the water column or deriving nutrition through symbiotic relationships with photosynthetic or chemosynthetic microbes. Tribrachidium presented yet another body plan, a circular disc roughly 5 centimeters in diameter, marked by 3 raised, curving ridges arranged in a triradial pattern around a central point. This threefold symmetry has no direct parallel in the animal kingdom, and its function remains poorly understood. Some researchers have proposed that the ridges directed water flow toward a central feeding surface. Others suggest a passive suspension feeding strategy. What is clear is that Tribrachidium represents a body plan that did not survive into the Cambrian, one of many Ediacaran forms that appear to have left no descendants. The seafloor environment these organisms inhabited was itself structured in a way that would become unrecognizable after the Ediacaran. Across the shallow marine shelves, microbial mats formed continuous, layered sheets across the substrate. Dense communities of cyanobacteria and other microorganisms that carpeted the sediment surface in a way that, today, is largely prevented by the bioturbating activity of burrowing animals. In the Ediacaran, no such animals existed. The seafloor was undisturbed. Sediment layers accumulated in fine, undisturbed laminations. The microbial mat surface provided both a stable substrate and, likely, a source of nutrition for the organisms that moved across or attached to it. This undisturbed seafloor is one of the most significant characteristics of the Ediacaran world. Without burrowing organisms to mix and oxygenate the sediment, the substrate remained chemically stratified, oxic at the very surface, but anoxic within millimeters of depth. The absence of predation is equally important. Current fossil evidence reveals no organisms in the Ediacaran biota that possessed structures clearly adapted for capturing and consuming other macroscopic organisms. There were no jaws, no claws, no grasping appendages. The ecosystems of this period operated without the dynamic of predator and prey that would come to define nearly all animal communities thereafter. Some organisms did move. Trace fossils, preserved tracks, trails, and burrows left by organisms moving through or across sediment begin to appear in the Ediacaran record at approximately 560 million years ago. These traces are simple, shallow, horizontal impressions consistent with slow movement across a matte surface, likely produced by organisms with muscular bodies capable of directed locomotion. The identity of the trace makers is uncertain in most cases, but their existence confirms that motility, the capacity for self-directed movement, had already evolved by the Middle Ediacaran, at least 20 million years before the period's end. Among the more structurally complex Ediacaran organisms was Cambrian, recovered from sediments approximately 555 million years old in both Russia and Australia. Cambrian was a bilaterally symmetrical organism with a distinct front and rear orientation, an external structure interpreted by some researchers as a flexible but somewhat rigid covering, and evidence in the form of associated scratch marks in sediment that it used a rasping organ to graze on microbial mats. If this interpretation is correct, Cambrian represents the earliest known evidence of an organism actively feeding on substrate material using a specialized structure, placing it in a possible ancestral or closely related position to the mollusks. Its body length reached approximately 15 centimeters, making it one of the larger mobile organisms of its time. Toward the final stages of the Ediacaran, roughly between 560 and 539 million years ago, the fossil record shows a gradual shift. The classical frondose and discoidal forms, Charnia, Tribrachidium, and their relatives, begin to disappear from the sedimentary record. In their place or alongside them appear organisms with features more consistent with the early animal body plans that would proliferate in the period that followed. Small, simple burrows become more common. Evidence of more active movement accumulates. The microbial mat ecosystems that had dominated for hundreds of millions of years begin to show signs of disruption as organisms capable of moving through and disturbing the substrate become more prevalent. The causes of this transition are not fully resolved. One hypothesis attributes the change partly to rising oceanic oxygen concentrations, which by the late Ediacaran may have reached levels sufficient to support higher metabolic rates, and the development of more complex tissue architectures. Another factor may have been the evolution of predation itself, the emergence, near the Ediacaran Cambrian boundary, of organisms capable of consuming other organisms, which would have imposed new selective pressures on the entire ecosystem. The disappearance of the classic Ediacaran forms may reflect competitive exclusion, predation pressure, environmental change, or some combination of these forces acting across millions of years. What the Ediacaran period establishes, above all, is a foundation. For approximately 96 million years, Earth maintained a biosphere of macroscopic, multicellular life operating entirely without the ecological structures. Predation, skeletal mineralization, active burrowing, complex food webs that would define all subsequent animal ecosystems. The organisms of this period were not primitive in a dismissive sense. They were the product of billions of years of evolutionary history, and they occupied and shaped their environments with a coherence and stability that persisted across geological timescales. They were, simply, different, a first expression of complex life operating under conditions and by strategies that the planet would not sustain again. By 539 million years ago, the sedimentary record that had preserved these forms so clearly begins to change. The laminated microbial substrates give way to disturbed, bioturbated sediments. The distinctive impressions of Ediacaran bodies become rare and then absent. The oceans were entering a new state, one in which the rules governing survival, reproduction, and ecological interaction were being rewritten at a pace the planet had not previously experienced. At 539 million years ago, the transition recorded in sedimentary sequences across every major continent marks one of the most consequential boundaries in the history of life. The chemical and physical signals are clear: carbonate isotope excursions, shifts in sediment texture, the abrupt appearance of mineralized fossil material where none had existed before. Within a geologically brief interval, estimates range from 13 to 25 million years, the oceans were populated by a diversity of animal body plans that would form the architectural basis of nearly all animal life that followed. This is the Cambrian explosion, and its causes remain among the most actively debated questions in evolutionary biology. The conditions enabling this transition had been accumulating for tens of millions of years. Oceanic oxygen concentrations continued to rise through the late Ediacaran and into the early Cambrian, reaching levels that current models suggest were sufficient to support the sustained aerobic metabolism required by active muscular animals. Simultaneously, the chemistry of shallow seawater, particularly concentrations of calcium and carbonate ions, had reached a threshold that made biomineralization energetically viable. For the first time in Earth's history, organisms could construct and maintain hard, mineralized structures, shells, plates, spines, and calcified frameworks. That would come to define animal anatomy. The earliest Cambrian fauna preserved in assemblages, such as the Fortunian small shelly fossils, were already diverse. These were not transitional experiments in isolation. They were the visible leading edge of a radiation that was proceeding rapidly across multiple animal lineages simultaneously. Within 20 million years of the period's opening, the Burgess Shale of what is now British Columbia, Canada, deposited approximately 508 million years ago, preserves a marine community of extraordinary complexity. Soft tissues, digestive systems, eyes, gills, and limbs are recorded in exceptional detail, providing a window into an ecosystem that was already mature and ecologically structured. Among the dominant predators of this early ocean was Anomalocaris canadensis, an organism reaching lengths of up to 1 meter, making it by far the largest known animal of its time. Its body was elongated and laterally compressed, propelled by a series of overlapping lateral lobes that functioned as undulating fins. At its anterior end, 2 segmented grasping appendages flanked a circular, tooth-fringed oral disc capable of closing with sufficient force to crush lightly mineralized prey. Compound eyes, preserved in some specimens as isolated structures, were composed of an estimated 16,000 individual lenses, a visual apparatus comparable in resolution to that of modern dragonflies, and almost certainly capable of detecting motion and form with precision. Anomalocaris was an active pursuit predator in a world where most other organisms had only recently acquired the capacity to move quickly at all. It belonged to a broader group known as the Anomalocaridids, and its existence illustrates a fundamental principle of the Cambrian transition, the emergence of predation as an ecological force. Once organisms capable of actively hunting other organisms appeared, the selective pressure on prey species intensified dramatically. Shells, spines, and defensive body architectures proliferated. Movement became faster. Sensory systems became more sophisticated. The evolutionary feedback between predator and prey, an arms race measured not in years but in millions of generations, had begun, and it would not cease. On the seafloor beneath the water column where Anomalocaris hunted, trilobites had become the most numerically abundant and morphologically diverse group of animals. In the Cambrian Ocean, trilobites were arthropods, members of the same broad lineage that includes modern crustaceans, insects and spiders, and their bodies were divided into 3 longitudinal lobes, a central axial lobe flanked by 2 pleural lobes from which the group takes its name. A calcite exoskeleton provided structural support and protection. The head shield, or cephalon, bore compound eyes in most species, some of the earliest and most anatomically sophisticated eyes preserved in the fossil record. Trilobite species ranged from less than 1 millimeter to over 70 centimeters in length. They occupied nearly every available marine habitat. Some were active swimmers in the water column, others were benthic crawlers, moving across sediment surfaces, and some are inferred to have burrowed into soft substrates. Their feeding strategies were equally varied. Deposit feeding, filter feeding, and predation on smaller organisms have all been proposed for different species based on morphological evidence. Over the course of the Cambrian and into subsequent periods, trilobites would undergo repeated radiations, producing thousands of species across hundreds of millions of years of geological time. The Cambrian seas also contained the earliest representatives of body plans that persist to the present day. Pichia gracilans, recovered from the Burgess Shale, is among the earliest known chordates, organisms possessing a notochord, the structural precursor to the vertebral column. It was a small, blade-shaped swimmer approximately 5 centimeters long, moving through the water column by means of sequential muscle contractions along its body. Its existence at 508 million years ago places the origin of the chordate lineage, the lineage that would eventually produce fish, amphibians, reptiles, birds and mammals, firmly within the Cambrian. Wiwaxia corrugata added another layer to this ecosystem's complexity. Covered in overlapping sclerites, mineralized scales, and bearing dorsal spines for defense, it moved slowly across the seafloor, rasping at microbial films and organic detritus with a specialized feeding structure. Opabenia regalis, with 5 eyes arranged across its head, and a flexible frontal proboscis, tipped with a grasping claw, represented a body plan with no living relatives. One of several Cambrian organisms whose precise phylogenetic relationships remain unresolved despite detailed anatomical study. The diversity of body plans present in the Cambrian fossil record led some researchers to propose that the period saw the establishment of more fundamental anatomical blueprints, phylum-level body plans than exist today. Current analysis is more conservative, suggesting that many Cambrian forms represent early, stem group members of lineages that are still extant, rather than entirely separate evolutionary experiments. Nonetheless, the morphological range preserved in sites like the Burgess Shale and the slightly older Chengjiang biota of Yunnan Province, China, reflects a genuine and rapid expansion of animal form that has no parallel subsequent history of life. The Cambrian Ocean was not uniformly populated. Reef-like structures, not coral reefs, but mound-shaped accumulations built by archaeoceaths, an extinct group of sponge-like filter feeders formed in shallow, warm, carbonate-rich waters during the early Cambrian. These structures created three-dimensional habitat complexity on the seafloor, providing shelter, attachment surfaces, and feeding opportunities for a range of associated organisms. By the Middle Cambrian, archaeoceath reefs had largely collapsed, possibly due to oceanographic changes or competitive pressures. But the ecological principle they demonstrated, that reef-building organisms could structure entire communities around themselves, would be expressed repeatedly in the periods that followed. Beneath and within this expanding ecological structure, the chemistry of the ocean and atmosphere continued to shift. Carbon isotope records from Cambrian sediments show repeated excursions, rapid fluctuations in the ratio of carbon isotopes that indicate large-scale perturbations in the global carbon cycle. Some of these excursions correlate with episodes of elevated extinction within the Cambrian fauna itself, suggesting that the period, for all its biological innovation, was not one of uninterrupted stability. Several distinct Cambrian fossil assemblages show evidence of abrupt turnover, species-rich communities replaced by different communities over intervals that may represent as little as a few 100,000 years. By approximately 485 million years ago, the Cambrian period had drawn to a close. The oceans it left behind were fundamentally different from those of the Ediacaran, structured by predation, populated by mineralized animals, and organized into layered ecological communities, in which organisms interacted through competition, consumption, and mutualism, in ways that would persist and elaborate for hundreds of millions of years. The body plans established during this interval, arthropod, chordate, mollusk, echinoderm, brachiopod, would carry forward as the structural vocabulary of animal life on Earth. By 485 million years ago, the transition into the Ordovician period was registered, not as a sudden break but as a gradual continuation of processes already underway. Sea levels, driven by the absence of significant polar ice and the thermal expansion of oceanic crust at spreading centers, rose steadily across the early Ordovician, flooding low-lying continental margins and producing vast, shallow eperic seas that extended across what are now North America, Europe, and parts of Gondwana. These warm, sunlit, carbonate-rich waters would become the setting for the most dramatic expansion of marine biodiversity that the planet had yet produced. The event is recognized in the paleontological literature as the Great Ordovician Biodiversification Event. Over approximately 40 million years, the number of marine genera recorded in the fossil record increased by a factor of roughly 3, a magnitude of diversification that exceeded even the Cambrian explosion in terms of the number of distinct lineages produced. The ecological and evolutionary mechanisms driving this radiation were multiple and interacting: the expansion of shallow shelf habitat, increasing ecological complexity that opened new niches, evolutionary innovations in feeding and locomotion, and a global climate that for much of the period maintained warm and stable oceanic conditions across a section of the planet's surface. Reef systems expanded dramatically during the Ordovician. Unlike the archaeoceath structures of the Cambrian, Ordovician reefs were constructed primarily by stromatoporoids, sponge-like organisms with a dense layered calcareous skeleton, alongside tabulate and rugose corals, which make their first significant appearance in the fossil record during this period. These reef frameworks accumulated across shallow tropical shelves, creating habitat complexity. That supported diverse communities of bryozoans, brachiopods, echinoderms, and mollusks. In some regions, reef structures extended for hundreds of kilometers along ancient continental margins, their carbonate skeletons accumulating at rates sufficient to produce limestone deposits, tens of meters thick over millions of years. Among the most visually striking inhabitants of the Ordovician Ocean were the nautiloids, cephalopod mollusks that had diversified rapidly from their Cambrian origins into a wide range of forms. The orthoconic nautiloids, characterized by long, straight conical shells, produced some of the largest animals in the Ordovician Sea. Camaraceras, among the most frequently cited examples, is estimated to have reached shell lengths of between 6 and 9 meters, though the largest estimates remain debated due to fragmentary preservation. These were active predators, using a jet propulsion mechanism, expelling water from a muscular siphon to move through the water column and capturing prey with a ring of grasping tentacles surrounding a hardened beak. Their presence at the apex of Ordovician food webs imposed predation pressure on trilobites, brachiopods, and smaller cephalopods, driving continued evolutionary responses in defensive morphology across their prey communities. Trilobites, which had dominated Cambrian seafloors, remained abundant and continued to diversify through the Ordovician. New families appeared with increasingly elaborate morphologies. Some developed elongated spines projecting from their lateral margins, structures that likely served as predator deterrents by increasing effective body size and making swallowing difficult. Others evolved enrollment capability, the ability to curl into a tight ball with the calcite exoskeleton forming a complete protective enclosure. The visual systems of certain Ordovician trilobites reached remarkable complexity. Eglina and related genera possessed hypertrophied eyes with hundreds of individual lenses, suggesting adaptation to low-light or deep-water environments. The water column itself supported a distinct ecological layer populated by graptolites, colonial organisms composed of interconnected individuals housed within a shared organic skeleton. Graptolite colonies drifted through the open ocean, suspended by gas-filled floats, or entangled with floating organic matter, filtering microscopic food particles from the surrounding water. Their organic skeletons, compressed into thin carbon films, are preserved across vast stretches of ocean shale, and their rapid evolutionary turnover, with individual species persisting for relatively brief geological intervals before being replaced by new forms, makes them among the most precise biostratigraphic tools available for dating Ordovician sedimentary sequences. A single graptolite species might define a time interval of less than 1 million years within a continuous sedimentary succession. On and within the seafloor, the ecological structure of benthic communities had reached a level of complexity far exceeding anything the Cambrian had established. Brachiopods, filter-feeding organisms with two hinged shells superficially resembling bivalve mollusks, but representing an entirely distinct body plan, diversified into hundreds of genera, occupying virtually every available substrate type across the shallow shelves. Echinoderms, which include the ancestors of modern sea urchins, sea stars, and crinoids, produced an extraordinary array of body plans during the Ordovician, many of which did not survive the period. Stalked crinoids anchored themselves to hard substrates and extended feathered feeding arms into the current, straining organic particles from the water, a feeding strategy so effective that crinoid communities would persist with modifications for the next 400 million years. Burrowing activity in Ordovician sediments increased measurably compared to the Cambrian, producing a progressive disruption of the laminated seafloor structure that had characterized earlier periods. This bioturbation mixed sediment layers, oxygenated subsurface sediments, and fundamentally altered the chemical environment experienced by organisms living within and on the substrate. The ecological consequences were significant. As burrowing became more pervasive, the microbial mat ecosystems that had dominated seafloors since the Precambrian were progressively suppressed, replaced by a sediment structure more closely resembling that of modern marine environments. The Ordovician also recorded the first tentative colonization of terrestrial environments by photosynthetic life. Spore assemblages recovered from Ordovician sediments, particularly from sequences in Gondwana, indicate the presence of early land plants, likely resembling modern liverworts in their structural simplicity. These were not vascular plants. They lacked the internal transport systems required to move water and nutrients efficiently. Across significant distances, they were thin, low-growing mats of photosynthetic tissue, confined to moist environments near water margins. Their ecological impact on Ordovician landscapes was minimal in comparison to what would follow in later periods, but their presence established a biological foothold on land that would eventually transform the continents entirely. For approximately 40 million years, the Ordovician marine system sustained its expansion, its reef frameworks accumulating, its predator-prey dynamics intensifying, its ecological tiers multiplying in complexity. The oceans of this period represented a biological richness that the planet had never before achieved, a layered, interconnected system of organisms, extending from the sediment surface through the water column to the open ocean, each community shaped by and shaping the others through processes of competition, predation, and environmental modification. The conditions sustaining this system, however, were not permanent. Beneath the Southern Ocean, the continent of Gondwana was drifting steadily toward the South Pole. Ice was beginning to accumulate, and the stability that had permitted 40 million years of uninterrupted diversification was approaching its limit. The stability that had defined the Ordovician marine world did not erode gradually. The geological record indicates that the disruption, when it came, was rapid by any measure applicable to deep time, a sequence of events compressed into approximately 1 million years that dismantled ecosystems requiring tens of millions of years to construct. The late Ordovician mass extinction stands as the second largest in Earth's history, eliminating an estimated 85% of marine species and collapsing the elaborate ecological architecture that the Great Ordovician biodiversification event had produced. The mechanism was glaciation. As the Ordovician progressed, the supercontinent Gondwana, encompassing what would become Africa, South America, Antarctica, Australia, and the Indian subcontinent, continued its slow southward drift, carrying its landmass toward the South Pole. By approximately 445 million years ago, a significant portion of Gondwana was positioned over or near the polar region, providing the continental substrate necessary for large-scale ice sheet formation. Evidence for this glaciation is preserved in the rock record of what is now the Sahara Desert, striated bedrock surfaces, glacial dropstones, and tillite deposits that record the passage and retreat of ice across a landscape that today is among the hottest on Earth. The ice sheets that accumulated over Gondwana were extensive. Current estimates, derived from sedimentary and geochemical proxies, suggest that ice volumes during the peak of the late Ordovician glaciation may have approached or exceeded those of the Pleistocene glacial maxima, the most recent ice ages that shaped the modern landscape. As water was drawn from the oceans and locked into continental ice, global sea levels fell. Estimates of the magnitude of this fall range from approximately 50 to 100 meters, though some models suggest the drop may have been larger during peak glacial conditions. Across the shallow Epiric seas that had provided habitat for the majority of Ordovician marine life, this fall was catastrophic. Vast areas of productive seafloor were exposed as dry land. The warm, carbonate-saturated shelf environments that had supported reef communities, trilobite populations, and brachiopod assemblages across multiple continents were drained, their biological communities compressed into diminishing habitable margins, or eliminated entirely. The cooling of surface waters compounded the effects of habitat loss. Tropical and subtropical marine organisms, the majority of the Ordovician fauna, were adapted to the warm, stable thermal conditions that had persisted for tens of millions of years. As surface temperatures dropped and cold, oxygen-rich waters from polar regions began to circulate more extensively through the ocean system. Thermal stress was imposed across full latitudinal range of the marine biosphere. Reef-building organisms, which are among the most temperature-sensitive of marine communities, experienced rapid and widespread collapse. Stromatoporoid and coral reef systems, which had extended across tropical shelves for millions of years, contracted to remnant patches or disappeared entirely from the record. The extinction unfolded in two distinct pulses, a pattern recognized in fossil assemblages across multiple continents and confirmed by independent stratigraphic analysis. The first pulse coincided with the onset of glaciation and the initial fall in sea level, a wave of extinction that preferentially affected organisms in shallow shelf environments as their habitat contracted. The second pulse occurred during the subsequent deglaciation as the ice sheets melted and sea levels rose again. This second wave is in some respects counterintuitive. The return of shallow marine habitat might be expected to facilitate recovery. Instead, the rapid influx of cold meltwater, combined with oceanographic changes in circulation and chemistry, imposed a second round of stress on organisms that had already been severely reduced in diversity and abundance. Among the groups most severely affected were the graptolites, whose open ocean colonies were disrupted by changes in water column stratification and nutrient availability. From a peak diversity of dozens of concurrent species during the mid-Ordovician, graptolite diversity collapsed to a handful of surviving lineages. Trilobites, which had diversified extensively through the Ordovician, suffered losses at the family level, with numerous lineages disappearing entirely from the record at the extinction boundary. Brachiopods, reef-building corals, bryozoans, and echinoderms all recorded significant reductions in genus and species diversity, with some groups losing more than half their known genera within the extinction interval. The nautiloid cephalopods, including the large orthoconic forms that had occupied the apex predator role in Ordovician ecosystems, were also affected, though the group as a whole survived the extinction and would continue to diversify in subsequent periods. The selective pattern of extinction across different groups reflected differences in environmental tolerance, geographic range, and ecological specialization. Broadly distributed, ecologically generalized organisms tended to survive at higher rates than those confined to narrow environmental conditions or restricted geographic ranges, a pattern consistent with mass extinctions produced by rapid, large-scale environmental change. The carbon isotope record from this interval shows a significant positive excursion, an increase in the ratio of carbon-13 to carbon-12 in marine carbonates that is interpreted as reflecting a reduction in the burial of organic carbon, a change in ocean circulation, or both. Accompanying shifts in oxygen isotope ratios confirm the cooling of surface waters and the growth of polar ice. Together, these geochemical signals provide an independent line of evidence consistent with the sedimentological and paleontological record of glaciation and extinction, allowing researchers to correlate the biological crisis with its physical driver across geographically separated sections. Recovery from the late Ordovician extinction was not immediate. The early Silurian sedimentary record shows reduced diversity, simplified ecological communities, and the dominance of extinction survivors, organisms that had persisted through the crisis and now occupied ecosystems in which competitive and predatory pressures had been substantially reduced. Reef systems did not return to their Ordovician extent for millions of years. Many of the ecological niches, vacated by extinct lineages, remained unfilled for extended intervals, their eventual occupation dependent on the evolutionary radiations of surviving groups into new morphological and ecological territory. What the late Ordovician extinction demonstrates with unusual clarity is the fragility of even the most elaborate and long-established biological systems when confronted with sufficiently rapid physical change. The marine ecosystems that were dismantled had been built over 40 million years of continuous diversification. Their collapse required less than 1 million. The differential between the pace of construction and the pace of destruction is one of the defining asymmetries of the fossil record, repeated at intervals across the history of life on Earth. The oceans that persisted through the early Silurian were reduced in diversity, simplified in structure, and dominated by the survivors of the late Ordovician extinction. Recovery was not a single event, but a prolonged process, proceeding at different rates across different taxonomic groups and geographic regions. Sea levels rose again as the Gondwanan ice sheets retreated, reflooding continental shelves and gradually restoring the shallow marine habitats that extinction had contracted. Within approximately 10 to 15 million years of the extinction boundary, reef systems had begun to re-establish, graptolite diversity had partially recovered, and benthic communities were rebuilding the ecological complexity that the crisis had dismantled. But the most consequential development of the Silurian period was not occurring in the ocean at all. It was occurring on land. The continents of the Silurian were, by any modern standard, inhospitable. There was no soil in the developed sense, no structured mineral and organic matrix capable of retaining water, cycling nutrients, and supporting rooted plant communities. What existed instead was a combination of bare rock, weathered mineral sediment, and thin crusts of microbial and early photosynthetic life that had been accumulating since the Ordovician. Temperatures on exposed rock surfaces fluctuated widely between day and night. Ultraviolet radiation, unfiltered by the relatively thin Silurian ozone layer, struck the land surface with an intensity significantly greater than that experienced by modern terrestrial organisms. The physical challenges of surviving on land, desiccation, thermal stress, gravitational loading without the buoyancy of water were substantial, and the organisms that began to meet them did so through a series of incremental biological innovations, rather than any single transformative adaptation. The earliest vascular land plants appear in the fossil record at approximately 430 million years ago. These were small organisms, rarely exceeding a few centimeters in height, with simple dichotomously branching stems and no true leaves or roots in the modern sense. Cooksonia, among the earliest and most widely documented, consisted of slender, upright axes terminating in spore-bearing capsules. Its internal anatomy included a primitive vascular strand, a central column of cells specialized for conducting water upward through the plant body, which represents a critical innovation. Without the ability to transport water internally against gravity, plant height is constrained to the few millimeters over which water can move by diffusion alone. Vascular tissue broke that constraint, enabling plant bodies to extend upward into the air column, where light availability was greater, and competition for photosynthetic resources reduced. The outer surfaces of these early plants were covered by a waxy cuticle, a layer of polymerized lipid material largely impermeable to water. This cuticle prevented desiccation, allowing the plant to retain internal moisture, even in dry atmospheric conditions. Gas exchange, necessary for photosynthesis and respiration, was managed through small pores called stomata, openings capable of opening and closing in response to environmental conditions, balancing the need for carbon dioxide uptake against the risk of water loss. The simultaneous presence of vascular tissue, a protective cuticle, and stomata in early Silurian plants indicates that the suite of adaptations required for sustained terrestrial life had already been assembled, at least in rudimentary form, by the middle of the period. The ecological impact of even these small, structurally simple plants was measurable. Rite-like structures, rhizoids, and, in some species, more developed anchoring organs penetrated the mineral substrate, physically disaggregating rock surfaces and contributing organic material to the developing protosoil layer. As plants died and decomposed, they added carbon compounds to the substrate, altering its chemistry and increasing its capacity to retain moisture. fungal networks, which evidence suggests were already in symbiotic association with early land plants during the Silurian, further accelerated the breakdown of mineral substrate and the cycling of nutrients. The process of soil formation, one of the most consequential transformations in Earth's surface history, had begun, driven by the biological activity of organisms whose individual bodies were measured in centimeters. While plants were establishing their presence on the continental surface, the arthropods, the most diverse and adaptable animal group in the Silurian Ocean, were also beginning to extend their range onto land. The fossil record preserves evidence of early terrestrial or semi-terrestrial arthropods from Silurian deposits, including fragmentary remains of myriapod-like organisms and arachnid precursors. Pneumodesmus newmani, recovered from Scottish Silurian deposits approximately 428 million years old, is among the earliest known air-breathing land animals, a millipede-like organism whose preserved spiracles, the openings of a tracheal respiratory system, confirm that it was capable of extracting oxygen directly from the atmosphere rather than from water. The transition from aquatic to terrestrial respiration required significant physiological reorganization. Gills, which extract dissolved oxygen from water by passing it across thin, highly vascularized membranes, are ineffective in air. They collapse under their own weight and deprecate rapidly when exposed. Tracheal systems, by contrast, conduct air directly to tissues through a network of branching tubes, bypassing the need for a centralized respiratory organ and functioning efficiently at the small body sizes characteristic of early terrestrial arthropods. The evolution of tracheal respiration, combined with a waterproofed exoskeleton already present in marine arthropods, made the transition to land structurally achievable for this group without the need for entirely novel body architecture. The Silurian landscape these pioneers occupied was not uniform. Coastal and riparian environments, areas adjacent to water bodies where moisture availability was relatively high, provided transitional zones in which organisms could exploit terrestrial resources while remaining close to the aquatic environments from which they had originated. Inland areas, beyond the reach of regular moisture, remained largely barren throughout the Silurian, their colonization dependent on biological and physical processes that would require millions of additional years to reach sufficient scale. The early terrestrial biosphere was in this sense a marginal phenomenon, significant in its long-term consequences, but limited in its immediate spatial extent. In the ocean, the Silurian saw the continued recovery and reorganization of marine communities. Coral and stromatoporoid reef systems rebuilt extensively across tropical shelves, producing carbonate structures of comparable scale to those of the Ordovician. Jawed fishes, the gnathostomes, diversified significantly during the Silurian, with both the placoderms, characterized by bony armor plating covering the head and anterior trunk, and the early chondrichthyans, the cartilaginous fish lineage that includes modern sharks and rays, establishing their presence in the fossil record. The evolution of jaws, derived through the modification of anterior gill arches, represented a profound expansion of feeding capability, converting a passive filter feeding or suction feeding apparatus into a structure capable of grasping, biting, and processing a wide range of prey types. By the close of the Silurian, approximately 419 million years ago, the biological transformation of the land surface was still in its earliest stages. Plant communities remained low, simple, and confined to moist environments. Terrestrial animal diversity was limited to a relatively small number of arthropod lineages. But the processes set in motion during this period, the development of vascular plant architecture, the formation of protosoil, the establishment of tracheal respiration in terrestrial arthropods, had created the ecological and physical preconditions for a far more extensive and complex colonization of the continents in the periods that followed. The Devonian period opened onto a world in which the basic framework of vertebrate evolution was already established, but largely unrealized in its potential. Jawed fishes had appeared in the Silurian, and the oceans and freshwater systems of the early Devonian inherited a diverse, but still relatively modest assemblage of fish lineages. Over the 60 million years that followed, that assemblage underwent a radiation of extraordinary scope, producing body plans, feeding strategies, and ecological roles that transformed aquatic ecosystems from the shallow coastal shelves of the open ocean, and eventually directed the first vertebrate animals toward the land itself. The dominant vertebrates of the Devonian seas were the placoderms, armored fishes whose bony head and trunk shields hinged at the neck allowed the entire skull to rotate upward during feeding, dramatically increasing gape width. This mechanism, known as cranial kinesis, enabled placoderms to generate feeding strikes of considerable speed and force. The group diversified into more than 200 known genera occupying a wide range of ecological roles. Some were benthic bottom feeders with flattened bodies and crushing tooth plates adapted for processing hard-shelled invertebrates. Others were active mid-water predators with laterally compressed, streamlined bodies. Among all Devonian predators, Dunkleosteus terrelli has attracted the most sustained scientific attention. Estimates of its total body length, extrapolated from the preserved bony head shields, the only portions typically fossilized, range from 4 to 6 meters for large individuals, with some estimates reaching higher. Its oral apparatus was not composed of true teeth in the conventional sense, but of self-sharpening bony blades formed by the edges of the jawbones themselves, maintained in a perpetually sharp condition by the continuous wear between upper and lower surfaces. Biomechanical modeling of the jaw structure, based on muscle attachment sites preserved on the head shield, indicates that Dunkleosteus was capable of generating bite forces in excess of 4,400 newtons at the jaw tip, among the highest calculated for any fish. The jaw could open fully in approximately 20 milliseconds, generating a rapid pressure drop that drew prey into the oral cavity before contact was even made. Dunkleosteus was not an isolated apex predator, but one component of a diverse placoderm fauna that included large-bodied forms across multiple lineages. The existence of multiple large predatory species within the same Devonian ecosystems implies prey communities of sufficient abundance and diversity to sustain them, a measure of the overall biological productivity of Devonian marine environments. Invertebrate communities on Devonian reefs and shelves, built primarily by stromatoporoids and tabulate corals, continuing their recovery from the Ordovician extinction, provided the foundational ecological layer upon which this vertebrate diversity rested. The early sharks, chondrichthyans with cartilaginous skeletons, were present throughout the Devonian, though their fossil record is complicated by the poor preservation potential of cartilage compared to bone. Teeth, scales, and occasional fin spines provide the primary evidence for early shark diversity. Cladocellac, known from exceptionally preserved specimens in late Devonian deposits of Ohio, reached lengths of approximately 1.8 meters and possessed a streamlined fusiform body with large, broad-based fins, a morphology consistent with active, sustained swimming rather than the ambush strategies employed by some later shark lineages. Its teeth were multi-cusped and suited for grasping rather than cutting, suggesting a diet of fast-moving fish rather than large prey requiring dismemberment. The coexistence of large placoderms and early sharks in Devonian seas established a predator guild of considerable complexity. Prey organisms, smaller fishes, cephalopods, and large invertebrates were subject to predation pressure from multiple directions and at multiple size scales simultaneously. The evolutionary responses observable in Devonian fish faunas, increased swimming speed, enhanced sensory systems, the development of defensive spines in various actinopterygian and acanthodian lineages, reflect an arms race dynamic operating across multiple interacting lineages. Among the ray-finned fishes, the actinopterygians, the Devonian record preserves early representatives of a lineage that would eventually become the most species-rich vertebrate group on Earth. Early actinopterygians were small, typically less than 30 centimeters in length, with a covering of thick, ganoid scales and relatively simple fin arrangements. Their diversity in the Devonian was modest compared to what would follow in subsequent periods, but their presence in both marine and freshwater environments from an early stage reflects a fundamental ecological flexibility that would prove decisive in their long-term evolutionary success. The lobe-finned fishes, the sarcopterygians, occupy a position of particular significance in the Devonian record. This group, which includes the lungfishes and the coelacanths among its living representatives, possessed paired fins supported by a series of internal bones arranged in a proximal to distal sequence that directly prefigures the limb skeleton of terrestrial vertebrates. In several Devonian sarcopterygian lineages, this fin architecture became progressively more robust, and the musculature associated with the fins increasingly complex, producing appendages capable of generating thrust against a substrate rather than simply sculling through water. Tiktaalik rosier, recovered from late Devonian deposits of Ellesmere Island in the Canadian Arctic and dated to approximately 375 million years ago, represents one of the most informative transitional forms in the vertebrate fossil record. Its pectoral fins contained a recognizable humerus, radius, and ulna, the three bones that form the upper and lower arm in all terrestrial vertebrates, along with more distal elements interpreted as wrist-like structures. Its neck was partially mobile, a feature absent in most fishes, allowing independent head movement relative to the body. Its ribcage was robust enough to support body weight, partially against gravity. These features collectively indicate an organism capable of propping itself up on its forelimbs in shallow water or on soft substrate, a functional precursor to weight-bearing terrestrial locomotion. The environmental context for this transition was provided by the shallow, warm, seasonally variable freshwater and brackish systems that characterized much of the late Devonian continental interior. These environments, river floodplains, deltaic systems, shallow lakes and ponds, were subject to periodic drying, fluctuating oxygen levels, and the kind of physical complexity that rewarded organisms capable of moving across substrate, rather than solely through open water. The ability to breathe air, present in multiple Devonian sarcopterygian lineages, through the use of a lung-like structure derived from the swim bladder, provided a critical supplement to gill respiration in low oxygen conditions. By the close of the Devonian, approximately 359 million years ago, the transition from water to land for vertebrates had not yet been fully realized, but the anatomical and ecological foundations for it were firmly in place. The oceans and freshwater systems the period left behind structured by vertebrate predation at a level of complexity and intensity without precedent in Earth's earlier history. The fishes of the Devonian had not merely diversified, they had restructured the aquatic world, and in doing so had produced, almost incidentally, the lineage that would eventually leave it. The land surface that existed at the opening of the late Devonian was not empty, but it was sparse. Low-growing vascular plants had been extending their range across moist continental margins since the Silurian, and by the early Devonian, plant communities of modest structural complexity had established themselves across floodplains, river margins, and coastal lowlands across multiple continental regions. These were communities dominated by small, leafless or near-leafless forms, organisms relying on photosynthetic stems rather than broadleaf surfaces, rooted shallowly in substrates that had only begun to develop the chemical and physical properties of true soil. Then, beginning at approximately 385 million years ago, the scale of terrestrial plant life changed in a way that had no precedent in Earth's history. The development of arborescence, the capacity to produce a tall, self-supporting woody trunk, required the solution of two interrelated structural problems. The first was mechanical. A plant body extending several meters into the air column must resist gravitational loading and lateral wind forces without collapsing. The solution was the evolution of lignin, a complex organic polymer deposited within cell walls to produce woody tissue of exceptional compressive and tensile strength. Lignin-reinforced wood could support vertical structures of a scale impossible for earlier plant architectures. The second problem was hydraulic. A tall plant must transport water from soil to leaf surfaces many meters above the ground, against both gravity and the evaporative demand of photosynthetic tissue. The elaboration of vascular tissue, the xylem and phloem systems conducting water and nutrients through the plant body, provided the necessary infrastructure. Archaeopteryx, appearing in the fossil record at approximately 385 million years ago, was among the earliest and most ecologically significant of the arborescent plants. It combined a woody trunk, anatomically similar to that of modern conifers in its cellular organization, with broad, fern-like frond branches capable of intercepting light across a large surface area. Archaeopteryx reached heights of up to 10 meters, with trunk diameters of approximately 1.5 meters at the base. Its root system was extensive and deeply penetrating by the standards of earlier plants, anchoring the tree mechanically and accessing soil moisture and nutrients from depth. Evidence from its geographic distribution indicates that Archaeopteryx dominated forests extended across vast areas of the late Devonian tropics and mid-latitudes, from what is now North America and Europe to parts of Gondwana. The ecological consequences of forest establishment operated across multiple systems simultaneously. Root systems of the scale produced by Archaeopteryx and contemporaneous arborescent lycopsids and progymnosperms penetrated bedrock fractures and accelerated the chemical weathering of silicate minerals. Silicate weathering consumes atmospheric carbon dioxide, incorporating it into carbonate minerals that are eventually transported to the ocean and deposited in sediment. The global expansion of deep-rooted forests during the late Devonian therefore produced a measurable drawdown of atmospheric carbon dioxide. Estimates suggest CO2 concentrations may have fallen by as much as 50 to 90% over the course of the late Devonian, driving a significant cooling of global climate that would contribute to environmental instability by the period's close. The physical structure of forests also transformed the behavior of rainfall and rivers. Forest canopies intercepted precipitation and moderated its delivery to the soil surface, reducing erosion and stabilizing sediment. Root systems bound soil particles together, preventing the kind of mass erosion characteristic of unvegetated slopes. The organic material contributed by decomposing leaf litter and woody tissue accumulated in soil profiles, increasing water retention and nutrient availability. Rivers draining forested catchments carried finer sediment loads and behaved differently from those crossing barren terrain, producing the meandering, floodplain-building river systems whose sedimentary signatures begin to appear prominently in late Devonian rock sequences. The development of thick organic soil layers introduced a new biological challenge, however. Lignin, the structural polymer that made arborescent plants possible, was a novel organic compound for which the decomposer communities of the late Devonian were largely unprepared. The fungi and bacteria capable of efficiently breaking down lignin, the white rot fungi of the Basidiomycete lineage, had not yet evolved the enzymatic machinery to process it at scale. As a result, fallen woody material accumulated in vast quantities in late Devonian and subsequent Carboniferous environments, decomposing only slowly, if at all. This accumulation of undecomposed organic carbon had significant geochemical consequences, contributing to the drawdown of atmospheric CO2 and, over longer timescales, to the formation of the coal deposits that would be laid down in massive quantities during the Carboniferous. The forest floor and its margins supported an expanding community of terrestrial animals. Arthropods, already present on land since the Silurian, diversified rapidly in response to the new habitat complexity and food resources created by forest ecosystems. Decomposing leaf litter, fungal networks, and the structural complexity of root systems and fallen woody debris created ecological niches that had not previously existed. Mite and springtail-like organisms, evidence for which appears in early Devonian terrestrial deposits, were among the first animals to exploit decomposing plant material as a primary food source, a feeding strategy that would support enormous arthropod diversity in subsequent periods. Among the vertebrates, the tetrapods, four-limbed vertebrates descended from the lobe-finned fish lineages discussed in the context of the broader Devonian fish radiation, were making their first documented appearances in terrestrial or near-terrestrial environments during the late Devonian. Ichthyostega and Acanthostega, both recovered from late Devonian deposits of Greenland, dated to approximately 365 million years ago, possessed recognizable limbs with digits, Acanthostega bearing 8 digits on its forelimb, yet retained features including internal gills and a tail fin that indicate these animals remained substantially aquatic in their habits. Their limbs were likely used for maneuvering through shallow, vegetated water rather than sustained overland locomotion, but the structural transition from fin to limb was complete in anatomical terms. The late Devonian closed under conditions of increasing environmental instability. A series of extinction pulses, collectively referred to as the late Devonian extinction, or the Kelvasser and Hangenberg events, eliminated a significant proportion of marine species, particularly reef-building organisms, placoderm fishes, and many open-water invertebrate groups. The causes remain debated, with proposed mechanisms including global cooling driven by the CO2 drawdown, associated with forest expansion, oceanic anoxia resulting from the increased delivery of nutrients to the ocean via weathering, and bolide impacts. What is clear from the sedimentary and geochemical record is that the marine world of the late Devonian experienced repeated, severe disruptions that reduced its diversity substantially before the period's end. On land, the forests persisted and continued to expand. The transformation of the continental surface initiated by arborescent plants during the late Devonian was irreversible in any practical sense. The soil systems, hydrological regimes, and atmospheric chemistry altered by forest growth could not revert to pre-forest conditions without the complete removal of terrestrial plant life. The continents had been permanently changed, and the ecosystems that would develop on them in subsequent periods would do so on a foundation physically, chemically, and biologically that the first forests had constructed. The world that opened into the Carboniferous period inherited the transformed continental surfaces of the late Devonian, soils deepened by root systems, rivers stabilized by vegetation, and an atmosphere already measurably altered by the carbon drawdown of expanding forests. Over the 60 million years that followed, these processes continued and intensified, producing environmental conditions with no precise modern analogue, and ecosystems of a structural character that Earth has not replicated before or since. The fundamental driver of Carboniferous ecology was atmospheric oxygen. Current geochemical models, constructed from carbon and sulfur isotope records in sedimentary sequences, indicate that atmospheric oxygen concentrations rose substantially through the Carboniferous, reaching an estimated peak of approximately 30 to 35% by volume compared to the present level of 21% by the late period. The mechanism was the accumulation of undecomposed organic carbon in the vast coal swamp forests that dominated tropical and subtropical lowlands across the joined landmasses of Laurussia and Gondwana. As woody plant material was buried in waterlogged, anoxic swamp sediments faster than it could be decomposed, carbon was removed from the active biological cycle and locked into what would become coal seams. Each unit of carbon buried in this way represented a unit of oxygen left unreacted in the atmosphere, and over tens of millions of years the cumulative effect was a global oxygen concentrations significantly above modern levels. The forests themselves were structurally unlike any modern ecosystem. The dominant trees were not the gymnosperms or angiosperms that characterize present-day forests, but lycopsids, a group whose modern representatives are small, creeping ground plants rarely exceeding a few centimeters in height. In the Carboniferous, lycopsids produced arborescent forms of extraordinary scale. Lepidodendron and Sigularia, the best documented of these, reached heights of 30 to 50 meters with trunk diameters at the base of up to 2 meters. Their trunks were not woody in the conventional sense. The structural support was provided primarily by a thick, photosynthetically active bark rather than a dense core of lignified wood, and their growth strategy was fundamentally different from modern trees. They grew rapidly to their full height, produced a single terminal burst of spore-bearing reproduction, and then died, falling into the swamp waters below. The ground beneath these forests was perpetually waterlogged. Carboniferous coal swamps occupied low-lying tropical basins where the water table was at or near the surface, producing conditions of near-permanent inundation across vast areas. The absence of efficient lignin decomposers meant that fallen trunks accumulated in layers, compressed over geological time into the coal seams that define the stratigraphic record of the period, and that would power the industrial economies of the 19th and 20th centuries. The environment was warm, humid, and CO2 depleted relative to earlier periods, a dense oxygen-rich atmosphere pressing down on a landscape of standing water, towering spore-bearing trees, and circulating organic debris. Within this environment, the elevated oxygen concentration had direct and measurable consequences for animal physiology and maximum body size. Oxygen delivery in arthropods is accomplished through the tracheal system, a passive or semi-active diffusion network that conducts air directly to tissues. The efficiency of tracheal oxygen delivery decreases with increasing body diameter, imposing an upper size limit on arthropod body plans under modern oxygen conditions. Under Carboniferous oxygen levels, this constraint was relaxed, permitting the evolution of arthropods of a scale impossible today. Meganeura monyi, a griffin flea, a member of an extinct order related to but distinct from modern dragonflies, possessed a wingspan of approximately 65 to 70 centimeters, making it the largest known flying insect in Earth's history. Its flight mechanics, reconstructed from wing venation patterns, indicate an active aerial predator capable of sustained flight and rapid maneuvering. Its prey almost certainly included other large insects and potentially small vertebrates. The air of the Carboniferous swamp forests was populated by flying arthropods at scales that the post-Carboniferous atmosphere could not sustain, and the ecological dynamics of aerial predation and prey evasion operated at body sizes with no modern equivalent. On the forest floor, Arthropleura, a myriapod related to modern millipedes, reached body lengths of up to 2.5 meters and widths approaching 50 centimeters, making it the largest known terrestrial invertebrate in Earth's history. Its fossil trackways, preserved in Carboniferous sediments across Europe and North America, record an animal moving across substrate with a stride pattern consistent with relatively rapid locomotion for its size. Gut content evidence and tooth morphology suggest Arthropleura was primarily a herbivore, feeding on the abundant spore-bearing plant material of the forest floor, though some researchers have proposed omnivorous habits. Its size, like that of Meganeura, is most parsimoniously explained by the elevated oxygen concentrations of its environment. The vertebrate fauna of the Carboniferous swamps was dominated by early tetrapods, four-limbed vertebrates that had diversified significantly from the transitional forms present in the late Devonian. Two major lineages had by this point diverged: the amphibians, which remained tied to water for reproduction due to their gelatinous, desiccation-prone eggs, and the amniotes, whose evolution of a shelled, internally membraned egg capable of surviving in terrestrial conditions represented a decisive biological innovation. Among the amphibians, forms such as Eriops reached lengths of approximately 2 meters, robust, heavily built animals with broad skulls and interlocking teeth suited for capturing large prey. Temnospondylls, a diverse amphibian group, occupied both aquatic and semi-terrestrial niches across the coal swamp ecosystem, their flattened bodies and laterally placed eyes consistent with ambush predation in shallow water. The earliest amniotes, by contrast, were smaller and more lightly built, lizard-like in general appearance. But their reproductive independence from standing water allowed them to exploit habitats beyond the swamp margins that amphibians could not consistently access. The amniote lineage had already begun to diverge into the two major branches that would define vertebrate evolution for the remainder of the Paleozoic and beyond. The synapsids, the lineage that would eventually produce mammals, and the reptiles, including the lineage that would lead to dinosaurs, birds, and crocodilians, were both present in the Carboniferous fossil record, though at this stage they were small, ecologically marginal components of ecosystems dominated by amphibians and invertebrates. Toward the close of the Carboniferous, global climate began to shift. The supercontinent Pangaea, assembled through the progressive collision of Laurussia and Gondwana, altered atmospheric circulation patterns and reduced the penetration of moisture into continental interiors. Glaciation expanded across Gondwana. The warm, humid tropical coal swamps that had dominated the landscape for tens of millions of years contracted and fragmented, a process known as the Carboniferous rainforest collapse, recorded in the sedimentary record at approximately 307 million years ago as an abrupt transition from coal-bearing swamp deposits to drier, red-bed sedimentary sequences. Forest communities were isolated into refugia separated by increasingly arid terrain, and the ecological connectivity that had characterized the coal swamp biome was severed. The consequences for biodiversity were significant. Amphibian diversity declined as their swamp habitats contracted. Many large arthropod lineages diminished or disappeared entirely as oxygen levels began their long decline toward the end of the Carboniferous and into the Permian. The amniotes, whose terrestrial adaptations made them less dependent on the specific conditions of the coal swamp, survived the transition in greater proportional diversity, and into the drier, more climatically variable world of the Permian they would expand. The world that opened into the Permian period was drier, cooler in its higher latitudes, and structurally more varied than the coal swamp landscapes that had preceded it. The supercontinent Pangaea had reached its maximum assembly, a single landmass extending from pole to pole and spanning nearly half the planet's circumference at equatorial latitudes. This configuration had profound consequences for global climate. The vast continental interior, insulated from the moderating influence of ocean moisture by sheer distance from any coastline, experienced temperature extremes of a magnitude unprecedented in the preceding periods. Seasonal variation was amplified. Monsoon systems of continental scale swept moisture across coastal regions, while interior basins remained persistently arid. The coal swamps were gone, replaced across much of the tropical and subtropical landmass by desert, semi-arid scrubland, and seasonally dry floodplain environments. Into this transformed landscape, the amniotes, whose reproductive and physiological independence from standing water had positioned them advantageously at the close of the Carboniferous, expanded and diversified with a speed and breadth that restructured terrestrial ecosystems entirely. The two major amniote lineages that had diverged in the Carboniferous now elaborated into ecologically dominant faunas, and for the duration of the Permian, it was not the reptile lineage, but the synapsids, the lineage ancestral to mammals, that produced the dominant large-bodied terrestrial animals on Earth. The earliest prominent synapsids of the Permian belonged to a group known as the polyxores, a grade of animals that, while not forming a natural evolutionary grouping in the modern cladistic sense, shared a broadly similar level of anatomical organization. Among them, Dimetrodon is the most recognizable. Reaching body lengths of up to 3.2 meters, Dimetrodon was the largest terrestrial predator of the early Permian, and its anatomy reflects adaptations for active pursuit or ambush of substantial prey. Its skull was deep and laterally compressed, bearing differentiated teeth, a feature technically termed heterodonty, with large, prominent canine-like teeth at the front of the jaw flanked by smaller cutting teeth. This differentiation, absent in most earlier tetrapods, whose teeth were uniform in size and shape, allowed more precise prey capture and processing. The most anatomically conspicuous feature of Dimetrodon was its dorsal sail, a structure formed by elongated neural spines projecting upward from the vertebrae, likely connected in life by a membrane of skin and connective tissue. The function of this structure has been debated extensively. Thermoregulatory hypotheses propose that the sail functioned as a surface area amplifier for absorbing or radiating heat, allowing Dimetrodon to warm its body more rapidly in the morning and extend its daily activity period relative to competitors lacking such a structure. Alternative proposals include sexual display or species recognition functions. Biomechanical modeling of heat exchange rates in sail-bearing versus non-sail-bearing animals suggests the thermoregulatory hypothesis is plausible, though it likely does not represent the sole function. By the Middle Permian, the pelycosaur-grade synapsids had been largely replaced by a more derived group, the therapsids, whose anatomical organization represented a substantial step toward the mammalian body plan. Therapsid limbs were positioned more directly beneath the body, rather than sprawling laterally, reducing the lateral bending of the trunk during locomotion, and improving the mechanical efficiency of movement. Skull and jaw musculature was reorganized in ways that increased bite force and chewing efficiency. In the most derived therapsid lineages, evidence from bone histology suggests elevated growth rates consistent with at least partial endothermy. The physiological capacity to generate and regulate internal body heat, though full mammalian-grade thermoregulation, was likely not achieved until after the Permian. The Gorgonopsians were among the most ecologically significant therapsid predators of the Middle and Late Permian, ranging in body length from approximately 30 centimeters to over 3 meters in the largest genera. Gorgonopsians were pursuit predators with elongated skulls, reduced post-canine dentition, and dramatically enlarged upper canine teeth that in large species exceeded 15 centimeters in length. These saber-like canines were not used for chewing. Gorgonopsians, like most non-mammalian synapsids, likely swallowed large chunks of prey whole or in large pieces, but for delivering deep incapacitating wounds to prey animals. Their limb proportions and skeletal structure suggest they were capable of sustained locomotion at moderate speeds, a significant advance over the more sprawling gait of earlier large predators. Their prey included the dicynodonts, herbivorous therapsids that became the most numerically abundant and geographically widespread large terrestrial herbivores of the Permian. Dicynodonts ranged from small, burrowing forms the size of modern rodents to large-bodied genera approaching 3 meters in length. Most possessed a highly reduced dentition, in many species reduced to a single pair of upper tusks, with the remainder of the jaw covered by a keratinous beak, suited for cropping tough, fibrous vegetation. Their success across the diverse Permian landscapes of Pangaea, from equatorial semi-arid zones to higher latitude temperate environments, reflects a degree of ecological generalism and physiological flexibility that allowed them to exploit plant resources across a wide range of conditions. Alongside the Gorgonopsians and dicynodonts, the cynodonts, a therapsid group whose anatomical organization most closely anticipated the mammalian body plan, were present in the late Permian, though initially as small, ecologically subordinate members of faunas dominated by larger synapsid and reptilian forms. Cynotodont skulls show a secondary palate, a bony shelf separating the nasal passage from the oral cavity, allowing simultaneous breathing and chewing, a feature with significant implications for sustained aerobic activity and elevated metabolic rates. The reptile lineage was also diversifying through the Permian, though it remained less ecologically dominant than the synapsids on land. Paraeosaurs, Large, heavily built herbivorous reptiles with broad, flattened bodies and extensive dermal armor occupied herbivore niches in several Permian ecosystems, reaching body lengths of up to 3 meters. Mesosaurus, among the earliest known aquatic reptiles, inhabited freshwater environments in what is now South America and Southern Africa, its distribution across the South Atlantic, a body of water that did not exist in the Permian, providing some of the earliest biogeographic evidence for the former connection of Gondwanan landmasses. Early archosaurine reptiles, the lineage from which dinosaurs, crocodilians, and birds would eventually descend, were present but small and inconspicuous in late Permian faunas. The Permian also sustained large amphibians in environments where permanent water was available. Temnospondyls, which had persisted from the Carboniferous, produced large-bodied forms in the Permian. Mastodonsaurus and related genera reached lengths of 4 to 6 meters, with massive, flattened skulls and interlocking teeth, suited for capturing fish and smaller tetrapods in shallow aquatic environments. These animals occupied ecological roles broadly comparable to those of large modern crocodilians, and their persistence through successive periods of increasing aridity reflects the resilience of the temnospondyl body plan under a range of environmental conditions. For approximately 47 million years, the terrestrial and aquatic ecosystems of the Permian sustained this complex, synapsid-dominated fauna across the varied landscapes of Pangaea. The evolutionary trajectories established during this period, the progressive refinement of synapsid anatomy toward mammalian-grade physiology, the early diversification of archosaurine reptiles, the ecological dominance of dicynodont herbivores represented biological investments of enormous long-term consequence. But the conditions sustaining these ecosystems were not permanent, and the geological processes building toward the period's end would produce a disruption of a magnitude that neither the Permian fauna nor any subsequent assemblage of life on Earth has equaled. The Permian-Triassic boundary, preserved in sedimentary sequences across every major continent and in marine deposits spanning the full breadth of the ancient Tethys Ocean, is among the most precisely studied intervals in the geological record. The signal it carries is unambiguous. Within a stratigraphic interval representing, by current radiometric dating, approximately 60,000 years or less, the diversity of life on Earth collapsed to its lowest point in the preceding 300 million years. An estimated 96% of marine species and approximately 70% of terrestrial vertebrate species disappeared from the fossil record. The ecological structures built over the entirety of the Paleozoic era, reef systems, predator-prey hierarchies, soil communities, marine plankton networks, were dismantled with a thoroughness and speed that no subsequent recovery would fully replicate. The primary driver was volcanism on a scale that is difficult to contextualize against any modern reference point. In what is now the Siberian region of Russia, a volcanic system known as the Siberian Traps began erupting at approximately 252 million years ago, eventually producing a flood basalt province covering an estimated 7 million square kilometers, an area approximately the size of the European continent, with lava flows in places exceeding 3 kilometers in depth. Total magma volume estimates range from 1 to 4 million cubic kilometers. The eruptions were not a single event, but a prolonged series of pulses, sustained over a period of approximately 1 million years, though the most geochemically and biologically significant interval appears to have been considerably shorter. The immediate atmospheric consequences were severe. Volcanic outgassing released vast quantities of sulfur dioxide, which reacted with atmospheric water vapor to produce sulfuric acid aerosols capable of reflecting incoming solar radiation and suppressing surface temperatures on timescales of years to decades per eruptive pulse. More consequentially, over longer timescales, the eruptions released an estimated 10,000 to 30,000 gigatons of carbon dioxide into the atmosphere, a a figure that current models associate with a global mean temperature increase of between 8 and 10 degrees Celsius sustained over tens of thousands of years. The carbon isotope record from Permian-Triassic boundary sections worldwide shows a sharp, negative excursion at the extinction horizon, consistent with the introduction of isotopically light volcanic carbon into the ocean atmosphere system. The Siberian traps magma intruded extensively into sedimentary basins containing large deposits of coal, evaporites, and organic-rich shales. Thermal metamorphism of these sediments released additional greenhouse gases. Estimates suggest that contact metamorphism of coal-bearing sequences alone may have produced quantities of carbon dioxide and methane comparable to the direct volcanic emissions. Halogenated compounds, including chlorine and bromine-bearing gases produced by the heating of evaporite deposits, reached the stratosphere and contributed to the destruction of the ozone layer, exposing surface environments to elevated ultraviolet radiation, a stress documented in the Permian-Triassic boundary record by the presence of malformed and mutated spores and pollen into terrestrial sediments from this interval. In the oceans, the consequences of rapid atmospheric warming and CO2 increase were expressed through multiple interacting mechanisms. Ocean surface temperatures in tropical regions rose to an estimated 35 to 40 degrees Celsius, values at or beyond the thermal tolerance limits of most marine organisms adapted to the stable conditions of the late Permian. Simultaneously, the dissolution of additional CO2 into seawater drove ocean acidification, reducing carbonate ion concentrations and making the precipitation and maintenance of calcium carbonate shells and skeletons increasingly energetically costly. For organisms such as corals, brachiopods, echinoderms and foraminifera, whose hard parts were composed of calcium carbonate, the combination of thermal stress and acidification was lethal across vast areas of the ocean. Oceanic anoxia compounded these stresses further. As surface waters warmed, the solubility of oxygen in seawater decreased. Simultaneously, the thermal gradient between surface and deep waters intensified, reducing the density-driven, overturning circulation that normally delivers oxygenated surface water to the deep ocean. Evidence from trace metal geochemistry and biomarker compounds preserved in Permian-Triassic boundary sediments indicates that anoxic and oceanic conditions, oxygen-depleted waters containing toxic hydrogen sulfide, expanded dramatically through the water column, potentially reaching into the photic zone in some ocean regions. The presence of isoreneurethane, a biomarker produced exclusively by green sulfur bacteria that require both light and hydrogen sulfide, in boundary sediments from multiple ocean basins, is interpreted as evidence that hydrogen sulfide-bearing waters reached near-surface depths, directly poisoning marine organisms that had survived the thermal and acidification stresses. The collapse of marine ecosystems proceeded with a selectivity that reflects the differential vulnerabilities of different organism groups. Reef-building organisms, corals, stromatoporoids, and the calcareous sponges that had constructed Permian reef frameworks were effectively eliminated. Reefs would not return to the geological record in any substantial form for approximately 10 million years following the extinction boundary. Brachiopods, which had dominated benthic communities for hundreds of millions of years, were reduced to a small fraction of their Permian diversity. Fusolinid foraminifera, complex, large-bodied single-celled organisms with elaborate calcified shells that had been among the most abundant organisms in Permian shallow marine environments, disappeared entirely and permanently. On land, the extinction of terrestrial vertebrates was equally severe, though the record is less complete than the marine record due to the lower preservation potential of terrestrial environments. The Gorgonopsians, the dominant large predators of the late Permian, disappeared entirely at the boundary, leaving no descendants. Most dicynodont lineages were eliminated, though one lineage, the Lystrosaurus group, survived and would become numerically dominant in the early Triassic. the temnospondyl amphibians were severely reduced. Among the synapsids more broadly, the diversity that had characterized Permian faunas was compressed into a small number of surviving lineages. The recovery from the End-Permian extinction was the most prolonged of any mass extinction in the Phanerozoic record. Carbon isotope records remained disturbed for approximately 5 million years following the boundary, indicating persistent instability in the global carbon cycle. Marine diversity did not return to pre-extinction levels for an estimated 10 million years. Early Triassic fossil assemblages are characterized by low diversity, ecological simplicity, and the dominance of a small number of opportunistic taxa, most notably Lystrosaurus on land, which in some early Triassic fossil beds accounts for more than 90% of individual vertebrate specimens recovered. The End-Permian extinction established, through elimination rather than transformation, the conditions under which the subsequent history of life would unfold. The synapsid-dominated ecosystems of the Permian were gone. The ecological space vacated by 96% of marine species and the majority of terrestrial vertebrate lineages was enormous, and its filling would require tens of millions of years and produce faunas of a character fundamentally different from anything the Paleozoic had generated. The world that emerged from the boundary was not a continuation of what had come before. It was, in the most literal biological sense, a new beginning. The earliest Triassic world was, by almost any ecological measure, impoverished. The sedimentary record immediately above the Permian-Triassic boundary preserves evidence of a biosphere operating far below its pre-extinction capacity. Low-diversity fossil assemblages, disrupted carbon cycling, and the repeated occurrence of microbial mats in marine environments that had not been dominated by microbial life since the Precambrian. The return of mat-forming microbes to shallow marine settings is itself a diagnostic signal. It indicates the collapse of the grazing and bioturbating animal communities that would normally suppress mat growth, leaving the seafloor temporarily resembling a world hundreds of millions of years older than it actually was. Recovery was not linear. The early Triassic carbon isotope record shows repeated negative excursions over a span of approximately 5 million years following the extinction boundary, each interpreted as a pulse of additional carbon input into the ocean atmosphere system, possibly from continued Siberian traps, volcanic activity, from the destabilization of seafloor methane hydrates, or from the oxidation of accumulated terrestrial organic matter. These pulses maintained environmental instability well beyond the initial extinction event, repeatedly interrupting the early stages of ecological recovery and preventing the sustained diversification that stable conditions would have permitted. On land, the dominant vertebrate of the earliest Triassic was Lystrosaurus, a dicynodont therapsid of modest dimensions, typically between 1 and 2 meters in length, with a barrel-shaped body, a keratinous beak, and a pair of upper tusks. Its survival of the End-Permian extinction, while most of its therapsid contemporaries perished, may reflect a combination of ecological generalism, burrowing behavior that provided thermal refuge, and a dietary flexibility allowing it to exploit the degraded plant communities of the post-extinction landscape. In multiple early Triassic fossil localities across what had been Gondwana and Laurasia, Lystrosaurus accounts for the overwhelming majority of vertebrate specimens, a dominance so complete that it represents one of the most dramatic examples of post-extinction ecological monopolization in the fossil record. Alongside Lystrosaurus, the temnospondyl amphibians persisted as significant components of early Triassic faunas, particularly in aquatic and semi-aquatic environments. Large-bodied temnospondyls, such as Capitosaurus, occupied predator roles in lakes, rivers, and floodplain water bodies. Their flattened skulls and broad gapes suited for ambush predation on fish and smaller tetrapods. Their persistence through the extinction likely facilitated by the relative buffering of aquatic environments against the most extreme temperature Spikes positioned them as ecologically important animals in the early recovery fauna, a role they would maintain for tens of millions of years into the Triassic and beyond. The Archosaur-line reptiles, the evolutionary lineage that would eventually produce dinosaurs, crocodilians and birds, diversified rapidly through the Triassic, exploiting the ecological vacuum left by the extinction of so many Permian vertebrate lineages. The early archosaurs split into two major groups recognizable from their ankle anatomy and posture: the Crurotarsi, whose descendants include modern crocodilians, and the Ornithodira, whose descendants include pterosaurs and dinosaurs. Through the early and middle Triassic, the Crurotarsal lineage, commonly referred to as Pseudosuchians, was numerically and ecologically dominant over the Ornithodiran lineage, producing a diverse array of large-bodied terrestrial predators and herbivores. Among the Pseudosuchians, the Etosaurs were heavily armored herbivores covered in rows of interlocking bony scutes, reaching lengths of up to 5 meters and occupying ecological roles broadly comparable to those of large modern herbivorous reptiles. The Rorsusuchians were large, active predators, some reaching 6 meters or more in length, with upright or semi-upright limb postures that allowed more efficient sustained locomotion than the sprawling gait of earlier reptiles. Phytosaurs, superficially resembling modern crocodilians in body plan and habit, occupied semi-aquatic ambush predator niches in rivers and lakes across much of the Triassic world. The first unambiguous dinosaurs appear in the fossil record at approximately 230 to 245 million years ago, depending on how the earliest archosaur material is interpreted. Eoraptor lunensis and Heterosaurus ischigualastensis, recovered from the Ischigualasto Formation of Argentina and dated to approximately 231 million years ago, represent early members of the dinosaur lineage, small to medium-bodied animals with fully erect limb posture, lightweight skeletons, and locomotor systems suggesting active, relatively fast movement. At this stage, dinosaurs were minor components of ecosystems dominated by pseudosuchian archosaurs and large temnospondyls. There is no indication in the early Triassic fossil record that dinosaurs would become ecologically dominant. Their rise, when it came, was contingent on events that had not yet occurred. The Triassic also witnessed the origin of the pterosaurs, the first vertebrates to achieve powered flight. The earliest pterosaurs appear in the fossil record at approximately 228 million years ago, already fully capable flyers with complex wing membranes supported by an elongated fourth finger, hollow bones reducing skeletal mass, and enlarged brains with expanded regions as associated with visual processing and balance. The absence of transitional forms between non-flying archosaurs and fully volant pterosaurs in the fossil record remains one of the most significant gaps in vertebrate evolutionary history. The origin of pterosaur flight is not yet resolved with the same anatomical clarity as the later origin of avian flight. In the marine realm, the Triassic saw the establishment of new ecological structures to replace those obliterated at the End-Permian. Ichthyosaurs, reptiles that had returned to the ocean from terrestrial ancestors, diversified rapidly through the Triassic, producing streamlined, dolphin-like body forms convergent with later marine mammals and modern toothed whales. Early Triassic ichthyosaurs such as Mixosaurus were relatively modest in size, but by the late Triassic, Shonisaurus popularis of Nevada had reached estimated lengths of 15 meters, making it among the largest marine animals of the Mesozoic. The evolution of live birth, documented in exquisitely preserved ichthyosaur specimens showing fetuses within the body cavity, removed the constraint of returning to land for egg-laying, allowing these animals to become fully pelagic in habit. The Triassic closed with another mass extinction event at approximately 201 million years ago. The End-Triassic extinction, associated with the eruption of the Central Atlantic magmatic province as the supercontinent Pangaea began to rift apart. This event eliminated the pseudosuchian archosaurs that had dominated Triassic terrestrial ecosystems, along with significant proportions of marine invertebrate and vertebrate diversity. The large temnospondyls were severely reduced. The conodonts, a group of jawless vertebrates that had persisted since the Cambrian, disappeared entirely and permanently. The extinction that closed the Triassic removed the ecological competitors that had suppressed dinosaur diversification for 30 million years. The cruotarsal archosaurs, the rhizoceans, atosaurs and phytosaurs were gone. The large temnospondyle predators of aquatic environments were largely eliminated. The ecological landscape of the earliest Jurassic was open in a way it had not been since the immediate aftermath of the End-Permian, and into that openness the dinosaurs, previously minor participants in Triassic ecosystems, began an expansion that would define the character of life on Earth for the next 135 million years. The ecological opening created by the End-Triassic extinction was filled with a speed and thoroughness that the fossil record documents clearly. Within the first 10 to 15 million years of the Jurassic, dinosaurs had diversified into the major lineages that would characterize the period: the large-bodied sauropods, the bipedal theropod predators, and the ornithischians, encompassing a wide range of herbivorous body plans. The transition from ecologically marginal Triassic survivors to dominant terrestrial vertebrates was not the product of any single biological innovation, but of the removal of competitors combined with the exploitation of ecological niches that had been vacated and remained available for colonization. The physical world into which this diversification proceeded was itself in transformation. Pangaea, the supercontinent assembled through the Paleozoic, had begun rifting during the Triassic, and through the Jurassic the separation of its constituent landmasses continued. The northern bloc, Laurasia, encompassing what would become North America, Europe and Asia, and the southern bloc, Gondwana, comprising South America, Africa, Antarctica, Australia and the Indian subcontinent, were separating along a widening seaway. Within Gondwana itself, rifting was initiating the separations that would eventually produce the South Atlantic. These tectonic movements altered ocean circulation, influenced regional climates, and created and destroyed land connections that shaped the geographic distribution of terrestrial faunas across the period. The climate of the Jurassic was, by current proxy evidence, significantly warmer than the present. Atmospheric CO2 concentrations are estimated to have been 3 to 8 times modern pre-industrial levels, suppressing the formation of polar ice caps and maintaining warm conditions at high latitudes. Tropical sea surface temperatures may have reached 30 to 35 degrees Celsius. The absence of significant polar ice meant that sea levels were substantially higher than today. Estimates suggest Jurassic sea levels may have exceeded modern levels by 100 to 200 meters, producing extensive shallow epicontinental seas across low-lying continental areas. These warm, shallow seas supported diverse marine communities of ammonites, belemnites, bivalves, and marine reptiles, while the continental interiors sustained the terrestrial ecosystems in which dinosaurs reached their greatest scale. The sauropod dinosaurs, long-necked, quadrupedal herbivores, are the defining biological phenomenon of the Jurassic in terms of body mass and ecological impact. The sauropod body plan, established in the early Jurassic from modest-sized ancestors, underwent progressive size increase through the period, driven by a combination of factors including the nutritional characteristics of available plant material, digestive physiology, and the mechanical advantages of large body size in processing bulk vegetation. By the late Jurassic, sauropods had produced the largest terrestrial animals in Earth's history, Brachiosaurus altithorax, known from the Morrison Formation of North America and dated to approximately 150 million years ago, reached estimated body masses of between 28 and 60 metric tons, with total body lengths of 20 to 26 meters. Its forelimbs were longer than its hind limbs, an unusual proportional arrangement among sauropods that elevated the anterior portion of the body. and extended the reach of its long neck upward, allowing access to vegetation at heights of 9 meters or more above the ground. Bone histology studies indicate rapid growth rates in juvenile Brachiosaurus, with individuals reaching near adult size within 10 to 20 years, a growth trajectory more consistent with elevated metabolic rates than with the slow, ectothermic growth patterns of modern large reptiles. Diplodocus carnegie, also from the Morrison Formation, reached total lengths of approximately 24 to 26 meters despite a body mass considerably less than that of Brachiosaurus. Its elongated neck and tail, combined with a relatively slender trunk, produced extreme linear dimensions without proportionally extreme mass. Its teeth were narrow and peg-like, arranged at the front of the jaw in a configuration suited for stripping foliage from branches rather than chewing it. Diplodocus almost certainly employed gastric processing, stones swallowed to assist mechanical breakdown of plant material in the digestive tract to compensate for the absence of chewing. The energetic demands of an animal of this mass required continuous feeding across most of the active day, and Morrison Formation paleoecological reconstructions suggest that sauropod herds exerted substantial pressure on the vegetation communities of their environments. The theropod lineage, bipedal dinosaurs with grasping forelimbs and serrated teeth, produced the primary terrestrial predators of the Jurassic. Allosaurus fragilis, the dominant large predator of the Morrison Formation ecosystem, reached body lengths of 8 to 12 meters and body masses estimated between 1.5 and 2.5 metric tons. Its skull was large relative to body size, but lightened by a series of fenestrae, openings in the bone, that reduced mass while maintaining structural integrity. Biomechanical analysis of its jaw musculature suggests that Allosaurus employed a hatchet-like feeding strike, driving the upper jaw downward into prey with considerable force rather than generating high bite forces through jaw closure alone. This strategy would have been effective against the large sauropods that shared its environment, allowing Allosaurus to inflict deep wounds on prey many times its own body mass. The vegetation sustaining these enormous herbivore communities was dominated by gymnosperms, plants reproducing by exposed seeds rather than enclosed fruits. Conifers related to modern Araucarius and podocarps formed the canopy layer of Jurassic forests, reaching heights of 30 to 40 meters in favorable environments. Cycads and Bennettitalians, palm-like plants with large, compound leaves, dominated the middle and lower vegetation layers. Tree ferns occupied moist, shaded environments. Flowering plants, the angiosperms that now virtually all terrestrial vegetation, had not yet diversified in the Jurassic, and the landscape lacked the broad-leafed deciduous phoreoses and flowering shrubs that characterize modern terrestrial ecosystems. In the skies above Jurassic landscapes, pterosaurs had diversified from the relatively modest forms of the late Triassic into a wide range of body, sizes and ecological roles. Rhamphorhynchus muensteri, with a wingspan of approximately 1 meters and a long, stiffened tail terminating in a diamond-shaped vein, was a coastal and open-water fish hunter whose preserved stomach contents confirm a diet of fish caught at or near the water surface. Pterodactylus, smaller and with a proportionally shorter tail, inhabited similar environments. These animals shared the aerial and coastal niches with the earliest birds. Archaeopteryx lithographica, known from the Solnhofen limestone of Bavaria, and dated to approximately 150 million years ago, represents the earliest widely accepted member of the avian lineage, combining unfeathered wings and a wishbone with retained ancestral features, including a toothed jaw and a long, bony tail. The Jurassic marine environment was no less productive than the terrestrial. Ammonites, cephalopod mollusks with coiled, chambered shells, diversified into hundreds of genera, their rapid evolutionary turnover making them the primary biostratigraphic tool for correlating Jurassic marine sequences globally. Ichthyosaurs, already established in the Triassic, reached peak diversity in the early and middle Jurassic before declining. Plesiosaurs, marine reptiles with broad bodies, 4 flipper-like limbs, and either long necks with small heads or short necks with large heads, diversified through the Jurassic into ecologically distinct lineages occupying different prey types and water depths. By 145 million years ago, as the Jurassic gave way to the Cretaceous, the dinosaurs dominated terrestrial ecosystems, and diverse marine communities of the period had established a biological template of extraordinary stability and productivity, one that would persist with modification for another 79 million years. The transition from the Jurassic into the Cretaceous was not marked by any mass extinction or sudden ecological reorganization. The dinosaur-dominated terrestrial ecosystems, the pterosaur inhabited skies, and the plesiosaur and ichthyosaur patrolled seas carried forward into the new period with their fundamental structures intact. What the early Cretaceous introduced was not disruption but elaboration, a progressive diversification of existing lineages, combined with the emergence of biological innovations whose long-term consequences would prove transformative well beyond the period itself. The breakup of Pangaea continued through the early Cretaceous at a pace measurable in the separation of faunal assemblages across different continental regions. The South Atlantic Ocean, opening as South America and Africa rifted apart along a widening mid-ocean ridge, was in the early Cretaceous still a narrow seaway, perhaps comparable in width to the modern Red Sea at its inception, gradually expanding toward the open ocean configuration it would achieve by the late Cretaceous. The separation of these landmasses progressively isolated the terrestrial faunas of South America and Africa from one another, initiating the independent evolutionary trajectories that would produce increasingly distinct dinosaur assemblages on each continent as the period advanced. In the northern landmasses, the separation of North America from Europe across the widening North Atlantic, and the increasing isolation of Asia, similarly partitioned dinosaur faunas into regional assemblages that diversified along independent lines. The result, visible in the early Cretaceous fossil record, was a global dinosaur fauna of greater geographic differentiation than that of the Jurassic, multiple regional ecosystems, each with its own characteristic predator and herbivore assemblages, connected by intermittent land bridges that permitted periodic faunal interchange without erasing the distinctiveness of regional lineages. Among the most significant biological developments of the early Cretaceous was the diversification of feathered dinosaurs, a radiation documented with exceptional detail in the Yixian and Jiufotang formations of Liaoning Province, northeastern China, deposited in a series of volcanic lake environments between approximately 130 and 120 million years ago. The Liaoning deposits preserve soft tissue impressions with a fidelity rarely achieved in the fossil record, revealing the presence of feathers, filamentous integumentary structures, and in some cases color-producing melanosomes that allow partial reconstruction of plumage patterns. Microraptor recovered from these deposits was a small dromaeosaurid dinosaur, approximately 77 centimeters in body length, bearing well-developed flight feathers on both its forelimbs and its hindlimbs, producing a four-winged configuration without direct modern analogue. Analysis of its feather geometry and aerodynamic properties suggests it was capable of gliding and possibly powered flight, though the precise mechanics of its locomotion remain debated. Its preserved stomach contents include fish, small birds, and lizards, indicating an active, versatile predator capable of exploiting resources across multiple habitat layers. Euanchuavis compsosaura and related early Cretaceous birds diversified into a wide range of body sizes and ecological roles, their skeletal anatomy showing progressive refinement of the flight apparatus, reduction of the bony tail, fusion of hand bones, and elaboration of the keeled sternum that anchors flight musculature, relative to the more archaic condition preserved in Archaeopteryx. By the early Cretaceous, the avian lineage had produced a genuine radiation of flying birds occupying coastal, aquatic, and forest environments across multiple continents, though all remained toothed and retained features of their dinosaurian ancestry, not present in modern birds. The non-avian theropods of the early Cretaceous included forms of considerable ecological and anatomical diversity. Spinosaurus aegyptiacus, known from North African deposits approximately 95 to 100 million years old, and therefore spanning the boundary into the late Cretaceous, represents one of the largest theropod dinosaurs yet documented, estimated body lengths ranging from 14 to 15 meters, with some estimates higher and a body mass potentially exceeding 7 metric tons. Its skull was elongated and conical, bearing numerous interlocking conical teeth suited for gripping slippery prey rather than cutting flesh. Isotopic analysis of its tooth enamel indicates a diet dominated by aquatic prey, and skeletal features including dense, pachyostotic bones reducing buoyancy and relatively short hind limbs are consistent with a semi-aquatic lifestyle, more dependent on water than any other large theropod in the record. The sauropod dinosaurs, which had achieved their greatest size in the Jurassic, continued to diversify in the early Cretaceous through the titanosaur lineage, a group characterized by wide-gauge limb posture, proculus vertebrae, and in many species the development of dermal ossicles, small bony nodules embedded in the skin. Titanosaurs would ultimately become the dominant sauropods of the Cretaceous, their geographic distribution eventually spanning every continent, including Antarctica. Some titanosaur lineages produced body masses potentially exceeding those of the largest Jurassic sauropods. Patagotitan majorum, from early to mid-Cretaceous deposits of Argentina, is estimated at approximately 69 metric tons, though the uncertainties in mass estimation for animals of this scale are considerable. The Ornithischian dinosaurs, the broad herbivorous lineage including the ancestors of later horned, plated and duck-billed forms, also diversified extensively through the early Cretaceous. Iguanodon bernissartensis, known from exceptionally complete specimens recovered from a coal mine in Bernissart, Belgium, in 1878, was a large bipedal to quadrupedal herbivore, reaching lengths of approximately 10 meters. Its hand bore a distinctive conical thumb spike, whose function has been interpreted variously as a defensive weapon, a tool for manipulating vegetation, or a display structure. Its dentition, batteries of interlocking, self-replacing teeth suited for processing tough plant material, represents a level of dental complexity exceeding that of most Jurassic ornithischians and prefigures the even more elaborate dental systems of the hadrosaurs that would follow. The most consequential biological event of the early Cretaceous, measured in terms of its long-term impact on terrestrial ecosystems, was the diversification of the flowering plants, the angiosperms. The earliest unambiguous angiosperm fossils appear in the record at approximately 13,000 to 125 million years ago, initially as small weedy plants occupying disturbed open habitats, river margins, floodplains, and other environments where rapid growth and early reproduction conferred competitive advantages. Fossil pollen assemblages show a progressive increase in angiosperm diversity and abundance through the early Cretaceous, with the group spreading from low-latitude disturbed habitats into a wider range of environments as the period advanced. The ecological consequences of angiosperm diversification were not immediately dramatic. Gymnosperm-dominated vegetation persisted as the dominant forest type through the early Cretaceous, but the biological infrastructure of the angiosperm radiation was establishing itself. The co-evolutionary relationships between flowering plants and their pollinators, insects, and eventually other animals, began generating a feedback system of mutual diversification that would accelerate through the late Cretaceous and into the Cenozoic, ultimately producing the most species-rich terrestrial ecosystems in Earth's history. By 100 million years ago, the early Cretaceous world had assembled an ecological complexity, surpassing that of the Jurassic in diversity of lineages, geographic differentiation of faunas, and the range of biological innovations in active deployment across terrestrial, aerial, and marine environments. The stage was set for the final, most diverse chapter of the dinosaurian age. The late Cretaceous represents the apex of the Mesozoic biosphere, the interval in which the biological systems established over the preceding 150 million years reached their greatest diversity, ecological complexity, and geographic extent. Continental separation had proceeded far enough by 100 million years ago that the major landmasses were approaching configurations recognizable in relation to the modern world, though significant differences remained. North America was bisected from north to south by the Western Interior Seaway, a shallow epicontinental sea connecting the Arctic Ocean to the Gulf of Mexico and dividing the continent into two landmasses for much of the late Cretaceous. Europe was an archipelago of low-lying islands separated by warm, shallow seas. India had detached from Gondwana and was drifting northward across the Tethys Ocean toward its eventual collision with Asia. These geographic configurations shaped the distribution of faunas with a precision that the more connected world of the Jurassic had not imposed. The climate of the late Cretaceous was among the warmest of the entire Mesozoic. Atmospheric CO2 concentrations remained elevated, polar ice was absent, and mean global temperatures are estimated to have been approximately 4 to 10 degrees Celsius above modern pre-industrial values. The Arctic and Antarctic regions supported forests of deciduous broadleaf trees adapted to the extreme photoperiod of high latitudes. Months of continuous daylight in summer followed by months of darkness in winter. Conditions that required physiological strategies quite different from those of lower latitude vegetation. Sea surface temperatures in tropical regions approached 35 degrees Celsius in some reconstructions, and the deep ocean was significantly warmer than today, reducing the thermal gradient that drives modern thermohaline circulation. Within this warm, geographically fragmented world, the dinosaur faunas of the late Cretaceous achieved their greatest taxonomic diversity. The ceratopsian dinosaurs, characterized by elaborate cranial ornamentation, including nasal and brow horns and a bony frill extending over the neck, diversified extensively across western North America and Asia. Triceratops horridus, among the last and largest of the ceratopsians, reached body lengths of approximately 8 to 9 meters and body masses estimated between 6 and 12 metric tons. Its skull, which could reach lengths exceeding 2.5 meters, was proportionally the largest of any known terrestrial animal. The three prominent facial horns, two above the eyes reaching lengths of approximately 1 meter, and one shorter horn on the nasal bone, have been interpreted through biomechanical and surface texture analysis as structures used in both intraspecific combat and defense against predators. Bone surface textures on ceratopsian frills show extensive vascular patterning consistent with a keratinous covering capable of color display, suggesting these structures served signaling functions in addition to any mechanical role. The hadrosaurs, duck-billed dinosaurs with elaborate dental batteries containing hundreds of continuously replacing teeth arranged in stacked columns, were the most numerically abundant large herbivores across much of the late Cretaceous of North America and Asia. Edmontosaurus regalis reached lengths of up to 13 meters and is known from bone bed deposits containing the remains of hundreds of individuals, indicating gregarious behavior and possibly migratory movement across seasonal environments. Parasaurolophus walkeri bore a hollow cranial crest extending posteriorly from the skull, its internal nasal passages forming a resonating chamber, whose acoustic properties, modeled computationally from CT scanned specimens, would have produced low-frequency vocalizations in the range of several hundred hertz, sounds capable of carrying over considerable distances through dense vegetation. Dominating the predator guild across Laramidia, the western landmass of divided North America, was Tyrannosaurus rex, the most extensively studied large theropod in the fossil record. Adult individuals reached body lengths of 12 to to 13 meters and body masses between 8 and 14 metric tons, with the largest known specimens approaching the upper end of this range. Its forelimbs were dramatically reduced relative to body size, each bearing 2 functional digits, but its hind limbs were massive, with femora exceeding 1.3 meters in length and cross-sectional bone geometry, indicating the capacity to support and accelerate substantial body mass. Bite force estimates derived from jaw muscle reconstruction and tooth root geometry range from 35,000 to 57,000 newtons, the highest calculated for any terrestrial animal, sufficient to crush bone and extract the marrow-rich contents of large prey carcasses. Isotopic analysis of Tyrannosaurus chemistry and growth ring counts preserved in bone histology sections indicate that individuals grew rapidly through adolescence, potentially adding several hundred kilograms per year during peak growth phases, before growth rates slowed dramatically in adulthood. Maximum lifespan estimates derived from growth ring counts in the most complete specimens range from 28 to 32 years. Evidence from multiple specimens preserves traces of soft tissue including scales, and in rare cases, possible integumentary filaments on specific body regions, though the extent of any feather-like covering in adult Tyrannosaurus remains unresolved. In the oceans surrounding the fragmenting continents, the late Cretaceous seas supported marine ecosystems of extraordinary productivity. The mosasaurs, large, fully aquatic lizards that had evolved from semi-aquatic ancestors in the early late Cretaceous, diversified rapidly to fill the ecological roles vacated by the declining ichthyosaurs, which disappeared entirely from the record at approximately 90 million years ago. Mosasaurus hoffmanii reached estimated lengths of 13 to 17 meters, its body propelled by a deep, laterally flattened tail and steered by 4 paddle-like limbs. Its jaw anatomy, including a secondary joint within the lower jaw, allowing it to flex and accommodate large prey, and its estimated bite force indicate a generalist apex predator capable of consuming large fish, ammonites, and other marine reptiles. Tylosaurus proctor, another large mosasaur, reached lengths approaching 14 meters and possessed a blunt, projecting rostrum anterior to the premaxillary teeth, interpreted as a ram used to stun or incapacitate prey for seizure. Prognathodon saturator bore robust, rounded teeth suited for crushing hard-shelled prey, including ammonites and turtles. This morphological diversity across mosasaur genera reflects a radiation into multiple feeding strategies within a geologically brief interval. The mosasaurs diversified from modest beginnings into dominant apex marine predators. Within approximately 20 million years, one of the more rapid ecological radiations in the vertebrate record. Above the late Cretaceous seas and coastlines, the pterosaurs reached their greatest body sizes. Quetzalcoatlus northropi from the Maastrichtian of Texas had an estimated wingspan of 10 to 11 meters, comparable to a small aircraft, and is estimated to have weighed between 200 and 250 kilograms, though these estimates carry substantial uncertainty given the fragmentary nature of the largest specimens. Its long, toothless jaws and elongated neck, combined with hind limb proportions suggesting efficient quadrupedal launch mechanics, are consistent with a terrestrial foraging strategy, stalking prey on open floodplains rather than plunge diving or surface skimming at sea. The flowering plants, whose diversification had begun modestly in the early Cretaceous, expanded dramatically through the late Cretaceous into forests, shrublands, and ground cover communities across all continents. By the Maastrichtian, the final stage of the Cretaceous, angiosperms had become the dominant vegetation type across much of the tropical and subtropical world. Their co-evolutionary relationships with insect pollinators and vertebrate seed dispersers, generating biological diversity across multiple taxonomic groups simultaneously. The insect fauna of the late Cretaceous, preserved in amber deposits from multiple regions, shows the presence of bees, ants, moths, and beetles with close relationships to modern flowering plants. a biological partnership already mature and productive 66 million years ago. By the final million years of the Cretaceous, the Maastrichtian world sustained dinosaur faunas of peak diversity across multiple continents, marine ecosystems dominated by mosasaurs and diverse shark lineages, skies patrolled by large pterosaurs and increasingly diverse birds, and continental vegetation restructured by the angiosperm revolution. It was, by every measurable biological metric, a world at the height of its Mesozoic elaboration. At 66 million years ago, in a geological instant by any measure applicable to Earth's history, the trajectory of life on this planet was redirected more abruptly and completely than at any point in the preceding 500 million years of animal evolution. The mechanism was an extraterrestrial impact, the collision of an asteroid approximately 10 to 15 kilometers in diameter with the shallow carbonate platform of what is now the Yucatan Peninsula of Mexico, at a location known today as Chicxulub. The energy released by this impact is estimated at approximately 100 teratons of TNT equivalent, roughly 1 billion times the yield of the nuclear weapon detonated over Hiroshima in 1945. The consequences, operating across multiple physical and chemical systems simultaneously, produced an extinction event that terminated the Mesozoic era and restructured the biosphere of Earth in ways whose effects persist to the present day. The immediate physical effects of the Chicxulub impact were regional in scale but globally significant in their secondary consequences. The impactor, traveling at an estimated 20 kilometers per second, vaporized on contact, releasing its kinetic energy in a pulse of thermal radiation and pressure that excavated a crater approximately 180 kilometers in diameter and 20 kilometers in depth within seconds. The target rock, a shallow marine carbonate and evaporite platform, was itself significant. Impact into sulfate-rich evaporite deposits released vast quantities of sulfur dioxide into the upper atmosphere, in addition to the carbon dioxide liberated by the vaporization of carbonate rock. Estimates suggest that between 100 and 500 gigatons of sulfur were injected into the stratosphere by the impact, a quantity sufficient to produce global sulfuric acid aerosol, loading orders of magnitude greater than any volcanic eruption in the subsequent geological record. The ballistic ejecta produced by the impact, rock and vapor expelled at velocities sufficient to reach the upper atmosphere and re-enter on suborbital trajectories, deposited thermal energy across the entire planetary surface as the material decelerated through the atmosphere. Current models indicate the re-entry heating of global ejecta may have raised atmospheric temperatures to several hundred degrees Celsius for an interval of several hours following the impact, sufficient to ignite surface vegetation across broad continental areas and expose unprotected organisms to lethal radiant heat loads. The evidence for this global fire pulse is preserved in a layer of soot and charcoal found at the Cretaceous Paleogene boundary in sedimentary sections worldwide, indicating the combustion of an estimated 70% of the global forest biomass within hours to days of the impact. The longer-term consequences were driven by the injection of particulate material, soot from wildfires, sulfuric acid aerosols, and fine silicate dust into the stratosphere, where residence times are measured in months to years, rather than the days characteristic of tropospheric aerosols. The resulting reduction in incoming solar radiation, commonly referred to as impact winter, suppressed photosynthesis across both terrestrial and marine environments for an estimated period of 1 to 2 years at full intensity, with partial suppression potentially extending for a decade or more as aerosol loading gradually diminished. The collapse of photosynthesis at the base of nearly all food chains propagated upward through trophic levels with a thoroughness that the fossil record documents in exceptional detail. In the oceans, the effects were compounded by rapid acidification. The sulfur dioxide injected into the atmosphere dissolved into ocean surface waters, producing sulfuric acid and driving a sharp drop in surface pH. Carbon dioxide released by the impact and by the subsequent decomposition of the enormous quantity of organic material killed by the event added to the acidification pressure. Calcareous plankton, foraminifera and coccolithophores, which formed the foundation of the open ocean food web and were responsible for a significant fraction of oceanic carbon cycling, experienced near total collapse across the boundary. The record of calcareous etonofossils at Cretaceous-Paleogene boundary sections shows an abrupt transition from diverse, abundant assemblages to near-zero diversity within a sedimentary interval representing, by sedimentation rate calculations, years to decades. The selectivity of the End-Cretaceous extinction reflects the mechanisms operating across this interval. Large-bodied animals with high absolute energy requirements were disproportionately vulnerable to the collapse of primary productivity. An ecosystem deprived of photosynthesis for 1 to 2 years could not sustain organisms requiring hundreds of kilograms of food per day. All non-avian dinosaurs, every lineage of large-bodied terrestrial dinosaur that had dominated continental ecosystems for 135 million years, disappeared from the fossil record at or within the boundary interval. No non-avian dinosaur species is known from Paleogene sediments anywhere in the world, despite intensive sampling across multiple continents and the fact that even a single surviving lineage would almost certainly have left a detectable trace in the subsequent record. The marine apex predators suffered equally complete losses. Mosasaurs, which had diversified into the dominant marine predators of the late Cretaceous within 20 million years, disappeared entirely at the boundary. The plesiosaurs, which had persisted since the Triassic, were eliminated. The large, diverse ammonite cephalopods, present in the ocean since the Devonian and having survived multiple previous mass extinctions, did not survive the Cretaceous-Paleogene boundary. Their last representative recorded in sediments within centimeters of the boundary layer itself. The pterosaurs, dominant aerial vertebrates for over 160 million years, left no survivors. The groups that survived the boundary shared characteristics that, in retrospect, reflect the specific stresses of the impact winter scenario: small body size, reducing absolute energy requirements to levels that could be met by surviving food resources, seeds, detritus, soil, invertebrates, was a consistent feature of terrestrial survivors. Ecological flexibility, allowing organisms to shift dietary sources when preferred food types became unavailable, conferred significant advantage. Freshwater ecosystems, buffered from some of the marine acidification effects, and sustained by allochthonous organic input from adjacent terrestrial environments, supported higher survival rates than open marine systems. Crocodilians, turtles, and small lizards survived at relatively high proportional rates compared to larger terrestrial reptiles. The avian dinosaurs, the birds, survived, representing the single dinosaur lineage to cross the boundary intact. Their survival likely reflected a combination of small body size, dietary flexibility, including seed eating and insectivory, and possibly behavioral flexibility that allowed exploitation of the degraded post-impact landscape. From this surviving lineage, the subsequent diversification of modern birds would eventually produce more than 10,000 living species, a radiation whose origins lie directly in the ecological opportunity created by the elimination of all competing non-avian dinosaurs. The mammals, present as small, ecologically subordinate animals throughout the Mesozoic, survived in multiple lineages across the boundary. Their subsequent diversification into the ecological roles, vacated by the extinction of non-avian dinosaurs, would define the character of the Cenozoic era, the age that followed, and would ultimately produce, across the subsequent 66 million years, the full diversity of modern mammalian life, including the primate lineage from which the species capable of reading and interpreting this record of Earth's deep past eventually emerged. The Chicxulub impact did not end life on Earth. It ended a particular configuration of life, one that had been building, elaborating, and diversifying for more than 500 million years from the quiet seafloors of the Ediacaran to the towering sauropods of the Jurassic to the tyrant predators of the Maastrichtian. What replaced it was not a continuation but a transformation, the opening of a new chapter written by the survivors of the most consequential single day in the history of complex life on this planet.